Alright folks, welcome back to topic 1.4. In this video we'll be discussing this idea of active transport. The previous video, again, we were talking about passive transport, and it's called passive transport because it doesn't require energy. And so if you had to guess what active transport was without looking at this header, then you might say, well, I know if I'm active, like I'm playing sports, or I'm going for a jog, or I'm doing something, I need to have energy for that. And that's a great thought, because active transport is this idea of transport that requires the use of energy for this to occur. Now, before I get into this, I want to apologize because this has already gone on longer than I planned. I originally planned on this only being a total of half an hour, let alone only passive transport taking a half hour. So I apologize for that. With that being said, I understand that the juniors last year struggled with this, and so I want it to be as in-depth and as detailed with this as I can for you. Additionally, you got time while you're sitting there in class anyway, so it's not the end of the world. You'll survive. With that being said, let's get into this idea of active transport. So again, we call this active transport because we need energy to move, because what we're doing is we're actually moving against or up a particle's concentration gradient. And so if we were to go back all the way to this diagram from our simple diffusion, this was simple diffusion because these were moving down a concentration gradient. We see them moving from a high to a low concentration, right? High to low. I had that mountain analogy. You start, you put a rock on top of a mountain, you push down, it's just going to go down naturally due to potential energy from gravity, right? No energy required besides that is already in the system. High to low concentration. But if we go to this idea of a low to high concentration, I went too far. We go this idea of low to high concentration. Now we need this energy, right? So if we're going to keep this mountain and rock or stone analogy, now imagine that you're standing at the base of the mountain and you're looking ahead with this stone in front of you, and you need to roll it all the way up this mountain. So it's going to take some energy for you to do that. We're moving against or up a concentration gradient, and so we need energy to do so. And the most common form of cellular energy is our good old pal adenosine triphosphate, ATP, produced by our wonderful friend the mitochondrion, the source of our, or the location of our cellular respiration. Active transport is performed by very specific proteins that are in the membranes. We talked generally about some channel proteins or carrier proteins, and these are more general, more generalized. Of course, not everything will be able to use the same channel proteins or the same carrier proteins, especially if it's a specific binding that's going to cause the shape shift. But when we're talking about active transport, we can get really, really specific to the point where they literally have names based off of what they're pumping. And so again, active transport here is performed by specific protein pumps that are embedded in our membranes. Our active transport, now we're going to get into our large and charged molecules. We'll see active transport done for large nonpolars, as well as maybe some ions that aren't exactly able to find some sort of channel protein. Sodium does have a channel protein, but sometimes it needs to be used in active transport. Or something very big like glucose and sucrose as well. And I like this analogy because, again, we're kind of looking at this mountain, working up/down a mountain here. The concentration gradient of the water is to move this way. In this case, it's from the bottom left to the top right of this image. But this truck is putting in energy to work against the flow of the water, is to work against the concentration gradient of the water to move up the river rather than follow down the river. It's like if you were to swim backwards in a lazy river. If you were to sit in a tube in a lazy river, you would just float along with the river all fine and dandy, but if you wanted to swim backwards, it might take you a little bit of time to get back there. And so this is what I was talking about when we can get very, very specific with our proteins. This is what we literally call a sodium-potassium pump. Take a guess why we call it a sodium-potassium pump. Well, I see it pumping both sodium and potassium. Clever right now. Anyway, what we see here is, in this situation, inside the cell, we have a low concentration of sodium, and a high concentration of potassium. That's inside the cell. Outside the cell, we have the opposite. We have a high concentration of sodium and a low concentration of potassium. What this image is depicting is that this sodium-potassium pump has 3 binding sites for our sodium ions. And so sodium will find its way into this cell, into this protein, and even though this is all kind of low concentration because particles move randomly, we saw that in the Colorado simulation, they will eventually find their way in. When this third potassium finds its way to our protein, at some point in time a molecule of ATP will also find its way there, and this is where we use that cellular energy, where our ATP is going to be broken down into a DP, adenosine diphosphate, because what is actually happening is this reallocation of phosphate onto the protein is going to cause this shape shift, similar to what we saw with a carrier protein. Right? In a carrier protein, we saw that this solute binding to this binding site will cause it to change shape from open outside to open inside, where it can continue down, and then after the release, it would find its way or shapeshift back into the open outside. And the presence of phosphate here is kind of doing that same thing. When phosphate is present, we'll see that this will break open or pop open towards outside of the cell. And again, because particles will move randomly, even though this is a low concentration and a high concentration of sodium, they'll move randomly and they'll find their way out of our protein. And what will also happen is the exact opposite from what we just saw with the sodium. Again, remember that outside of the cell is a low concentration of potassium and inside is a high concentration of potassium. But again, due to random movement, particles or ions of potassium will find their way and bind to their specific binding site here in our protein. And this second binding of potassium is going to cause yet another shapeshift of protein to the point where this phosphate can no longer hold its bond and will break off. And well, what do we see happen when this phosphate didn't exist? Well, we saw this protein open to the inside. And so same thing as we saw in steps 2 and 3, we see in steps 5 and 6. We see the protein is now changing shape to inside the cell. Our potassium can now randomly find its way towards outside of the cell. Again, this is called a sodium-potassium pump because we're pumping sodium and potassium. You might also see, I forget the name of it. It's fine. Just stick with sodium-potassium pump for now because I forget the name of it. It's all good. The wonderful diagram in your notes is, again, like I said, a wonderful diagram. It shows us in a very similar way. Step 1 is there in the top left, and it will follow this kind of clockwise configuration that our slides here also show it. There was a video here, but it was broken, and so I can input a video here after I record this, so you have a video here. And so this is just a quick little overview of these ideas of passive versus active transport. Again, these are passive transport here on the left because these are moving down a concentration gradient. They are not requiring energy because of it. These molecules are free to move where they need to, or where they randomly will even. It does not require energy, whether you have a carrier or channel protein or not. Either way, our purple balls here are moving down their concentration gradient. Our red triangles are moving down their concentration gradient just through a channel protein. And our blue balls are moving down their concentration gradient just through a carrier protein. And we have our active transport here on the right because now we have the movement of molecules against their concentration gradient, right? Remember that In this case, sodium is the little yellow balls here are being pumped out, and potassium, the little orange diamonds or squares that are rotating 90 degrees or 45 degrees, are getting pumped in, but they're moving against their concentration gradient, and so we need some sort of energy to help us do that. In this case, our energy is our ATP. And to save you the pain of having to sit through any more videos, we can be done here. We will talk about then this idea of membrane-assisted transport together later. But I don't want to have to make you sit here and do more of this, because I don't think either of us are really feeling like that right now. So hopefully this helps. I will of course leave these up for you so you have them. If you have any other questions, please, of course, you are more than welcome, and you know you're more than welcome to send me an email if you need to. I will do my best to help you. If you need office hours, please talk to me about that. I will love to help you with that as well. With that being said, thank you so much for listening, folks. I hope these were helpful, and I will see you all later.